evening, everybody. And thank you for coming out, and some of you might have had to swim in here tonight. I'm sorry, that wasn't the weather I ordered, but I'm grateful that you're here to spend the evening with us at a HudsonAlpha U night. And so for those in the room, welcome. For those that might be watching via the wonders of technology that is now working in the room, welcome to you too. I hope this will be a fun night. This is a combination of science and history tonight. We're going to take a journey together and tell some stories about incredibly brilliant scientists and the discoveries that they made that have really kind of catapulted genetics and what we know about DNA forward, kind of through the lens of history. And so we're going to go on a journey tonight through that. We've got a few props over here that if I can remember to, I'm going to pull to center stage as we tell those stories, and I promise the sewer cover will, there's a story for that. I promise they all connect, but it should be fun. So if anybody here doesn't know who I am, my name is Kelly East, and I have the pleasure of leading the education mission area here at HudsonAlpha and get to connect with learners of all ages, students, teachers, the public, and talk about DNA, why it matters, and how it impacts our lives. I feel like there's a lot of feedback. Pull it down some. But let's get started. So before we jump into Nobel Prizes and the people who have won Nobel Prizes in genetics, I think we need to start with the man who created the Nobel Prize itself. And that would be this gentleman right here, Alfred Nobel. So Alfred Nobel, let me introduce you to him a little bit. And I'm going to move this very authentic looking dynamite over here to the table. Don't worry, it's hollow. Alfred Nobel, he was a Swedish chemist, businessman. He was an inventor. He was born in Sweden, clearly. His dad was an inventor as well. But his dad was, at least early in his career, not terribly successful, ended up filing for bankruptcy, and moved his family, including young Alfred, they moved to Russia, where he had a fresh start, started doing inventions, working in the space of manufacturing, and started dabbling in explosives. And he was much more successful in that, and really young Alfred followed in his dad's footsteps and got very interested in developing tools and inventing things, and also playing around with explosives. So one of the things that Mr. Nobel is most well known for is he was the inventor of dynamite. He invented dynamite before his invention happened. The explosive that most people were using was a product called nitroglycerin, which worked well at blowing things up, but it had one kind of fatal flaw in that it sometimes blew up when you didn't want it to. It's not so great. And so Alfred Nobel, he figured out how to take nitroglycerin, but then ultimately mix it together with some other substances that would then make it more stable and only blow up when you are intending for it to blow up. And so he invented that, he patented that, and it became an immediate success with lots of utilities in oil mining as well as military and otherwise. He also developed another version of nitroglycerin that was actually a jelly-like substance that was even more powerful than dynamite. So he invented dynamite. That's what we need to know about him. But from that, he amassed quite a bit of wealth. He and a couple of his brothers, they created an oil company where they were going out and cultivating new locations for oil mining, and that led to quite a bit of wealth. In addition, he himself ran about 90 armament factories. So yeah, he had a lot of wealth. He also never married, and he didn't have any children. You can see where we're going, right? So when he passed away in 1896, he had quite a large bank account at the time. And so in his will, when they read his will in 1896, it explicitly laid out plans that the vast majority of his wealth was to be set aside and used to award a series of annual prizes that they would keep getting awarded into perpetuity, and that they would get awarded in these 5 categories, and they would be awarded to the individuals in each of those categories that in the previous year conferred the greatest impact to mankind. So that's where the Nobel Prize came from, and he said these were the 5 categories that he wanted to have prizes awarded in. So chemistry, physics, physiology or medicine, those are combined, literature, and peace. And he also explicitly said, well, he wanted those to be decided upon specific institutions. It was 3 institutions there in Sweden and then 1 in Norway that he said, these are the groups that I want awarding these prizes. That's who should make those decisions. The rules also went on to say that you couldn't win these awards if you were already passed away. You might have heard that. You can't posthumously be awarded a Nobel Prize. Although, fun fact, it has happened once. In 2011, there was a scientist who won the Nobel Prize for chemistry. No, I think it was physiology or medicine. And he actually passed away 3 days before it was announced. Yeah. And he was a brilliant scientist. He had discovered a type of immune system cell that was really important, and they let that one go because they said, look, when he was nominated, he was alive. And in fact, the Nobel Committee had no idea that he had passed away. And so that is one example of someone who was awarded one after death, but they were nominated before then. So these started being awarded. The first Nobel Prize winning ceremony happened on December 10th in 1901. It was exactly on it was on the 5th anniversary of his death, was with the first time that they were awarded. What else do we want to say about the Nobel Prizes? So you also cannot get a Nobel Prize as an organization with the exception of peace. So all of the other Nobel Prizes have to go to a specific person. Sometimes they go to multiple people, and we'll see examples tonight of that playing out, but it has to go to people. The Nobel Peace Prize is the only one that can actually be given to an organization. For example, the Red Cross has gotten the Nobel Peace Prize a couple of times. So the Nobel Prize, I imagine most people have heard of it, is widely regarded as kind of the highest honor in intellectual merit to be awarded a Nobel Prize. So when we think about Nobel Prizes and genetics, your brain, like mine, might go immediately to these gentlemen right here. On the screen behind me, this is James Watson and Francis Crick, who were the scientists, one American, one English, that are responsible along with other people. They did not do it alone, but they got the Nobel Prize for determining the structure of DNA, that DNA is a twisted ladder. It's certainly an incredibly important discovery. In fact, we still, at HudsonAlpha, we make a big deal every April that it's DNA Day. We celebrate it with students and teachers in our community. And part of that is honoring this particular discovery that happened at the end of May in 1953. But tonight, we're not going to dig into Watson and Crick, because I hope you've maybe heard some of their story before. We're going to talk about a few other scientists and discoveries that maybe you've heard less about. But before we jump into some of those stories, I also want to center us around, so 1901, this is when the first Nobel Prizes were awarded, to kind of look backwards from there and say, what did we know about DNA at that point? Who were the people and the players and the discoveries that had already happened? People that they didn't win a Nobel Prize because, well, Nobel Prizes weren't around when they were doing their work, or else they probably would have earned one. So we have to certainly talk about this gentleman right here. This is Gregor Mendel. Gregor Mendel was an Austrian biologist, also a religious abbot, who did some really famous scientific studies with pea plants in the monastery garden. He is the guy who really coined the term units of heredity. He was watching those pea plants grow and reproduce and was noticing that certain traits of those pea plants were being passed down in predictable patterns. So he started to figure out that, hey, there's this unit, there's this thing that's getting passed down from generation to generation that's guiding traits, guiding what these plants look like. But at that point, we don't know what DNA is, we don't know what those units are, but he really is, we often call him the father of genetics. So he did that work way back in 1865. And then also happening around that same period, there was this guy, Friedrich Miescher, who was doing some work as well. And Friedrich Miescher, he was Swiss in background, and he was really, really interested in figuring out what is happening inside of cells and what are the different chemicals, what are the components, what are the substances that make up our cells. And Miescher, along with many of the scientists we're going to talk about tonight, or at least the early ones, they were fascinated by proteins. And they really were convinced that proteins were the recipe book for life, that was the code, that proteins were going to unlock everything. And so he was studying and trying to figure out what are the proteins inside the cells, how is that guiding the recipe for life. You might be curious why there's a picture of a bandage up here. Things I was not going to put on my bookshelf over here. He did his research on infectious pus-filled bandages. Thank you. Those on the front row, you're welcome. Yeah, we're not doing that. But that was the product that he was using to study, and he was studying specifically, the white blood cells and stuff that you could get off of those bandages, and looking inside of there. And he was curious he recognized that there was something happening inside the nucleus of those cells that was peculiar and didn't behave like proteins were, that there's something else inside of those cells. Couldn't figure out what it was, but he noticed there was something there, and he called it nuclein. He also characterized it and said, there's something happening, I don't know what it is, but it seems to have a whole lot of phosphorus, and it seems to be really acidic. That was how far he got. Well, the A in DNA is acid, so he was absolutely describing DNA. And Friedrich Miescher is going to flow us right into the first person that I want to do kind of a deep dive into. And that is this gentleman right here, Albrecht Kossel. And I'm going to put back my dynamite. And we're going to get some cows. It'll make sense in a minute, I promise. So we're going to talk about Albrecht Kossel. And he lived in the 1800s and 1900s, and he was awarded the Nobel Prize in Physiology and Medicine in 1910. So that was within that first decade of Nobel Prizes being awarded. So Albrecht Kossel, he was born in the city of Rostock, which is modern-day Germany. At the time, it was not Germany. At the time, it was Prussia, which is part of the German Empire. But he was born in the city of Rostock. His dad was a incredibly wealthy businessman, banker, politician in the city. This was a family that had plenty of access to education and the kind of finest tutors and all of that. When it was time for him to go to college, he went off and decided to go study at the University of Strasbourg. And this is a map that doesn't look like Europe today. This is Europe in the 1800s. But right here, this is Strasbourg. And so he went to the University of Strasbourg, and notably, that university, when he was heading there, they were just off the cusp of being reestablished as a German institution. after the Franco-Prussian War. It had just kind of switched back sides, and they were rebuilding this university as a German institution. So he went there. He took some classes by a particular professor named Felix Seiler, which he'll come up here again in a minute. But he spent a couple of years there before he went back to the city of Rostock to finish his studying in college, which Rostock is somewhere up on the Baltic Sea. So he went back there. He studied medicine. He thought he was going to be a physician. He passed his medical boards, but was always fascinated by science. And so he never actually went and practiced medicine. When he finished in school and passed his medical boards, he decided to call up that professor back at the University of Strasbourg, Felix Seiler, and said, "Hey, can I get a job? I think I want to do science." And so he returned to that university and started working in that professor's lab. Do you know who also worked in that professor's lab? Friedrich Miescher, the bandages story? Yeah. So they were colleagues in this lab at the University of Strasbourg. And so Kossel also began to have an interest in figuring out what is happening inside of these cells in the nucleus. What are these non-protein components? And so that brings us to cows. So he took a different tactic of getting things to research. He didn't go the bandage route. Instead, he built a relationship with the local slaughterhouse. And so he got himself a steady supply of cow parts, right? That he could use as samples to research. And so he was looking at the cells of, initially, he was getting a whole bunch of cow pancreases sent to him, and he was looking at that and trying to figure out what was happening inside of that nucleus. And in his research, he very clearly articulated that nuclein subject that Miescher had coined, that term, that it is a substance that's made up of proteins and non-proteins. And we really need to figure out what these non-proteins are. But really, these guys were still much more interested in the protein component. They didn't quite figure out that this was the even more important bit, at least not yet. So before I kind of jump into what he discovered and how he contributed and why he got a Nobel Prize, I want to take a fast forward to 2024 and center us on what we know now is happening in the nucleus before we go back and talk about kind of how they kind of were helping get us there. So this is a super oversimplified picture of a cell. There's a lot more happening inside of a cell than what's pictured here, but just like Kossel, we care about the nucleus. So this is simplified. This is a cell, and we've got a nucleus here, and inside that nucleus is DNA. We know that now. It is full of DNA. So here's a picture of DNA, although this is a tiny, tiny fragment. If this was a human cell, there would be 3 billion of those latter rungs, so much, much larger than what's pictured. But inside our nucleus is our DNA. That really is the large function of the nucleus, is to be that protective, that place that our DNA is stored. And our DNA is the recipe for life, as we know now, that we have DNA and that there are sections of DNA that give us the recipe for specific functions. So we've got a piece of DNA, a stretch of DNA that tells our body how to do all the various things that it does, all the proteins it needs to make. It's the recipe for life. But our DNA, it never leaves the nucleus. It stays there where it is safe. So how on earth does DNA actually functionally code for life? How is that recipe used? And the way this plays out inside of our cells all the time is that those sections of DNA that code for a particular function, they end up getting copied into a different molecule called messenger RNA. Sometimes I'll use an analogy of a work site that if you are constructing a new building, you've got a master set of blueprints that has the instructions for everything, and then maybe you've got other types of smaller sections of blueprints that tell you how to do plumbing or electrical or you name it, and that you have those smaller working copies that give you information about one particular piece, that you make that copy and take that to the job site, but you're going to leave your master set of everything back where it's safe. And it's a similar kind of setup that happens inside our cells, where we have our DNA that stays in the nucleus, and that a particular section of it gets copied into a molecule of RNA. And what's unique about that RNA is it doesn't stay in the nucleus. It then can move out of the nucleus into the rest of the cell, where it is ultimately used as the recipe for building a protein, which is what most of our genes do, is they are a recipe for a particular protein. And then that is ultimately what goes out and builds life. And so this process of DNA to RNA to protein, we now call that the central dogma. It's a really important kind of conception of how DNA codes for life, that the DNA is in the nucleus, and then you have this RNA copy that makes protein. So we know all of this now. Way in the future. Kossel for sure did not. But what he did, and this is what he ultimately got the Nobel Prize for, was over the period of time from 1885 to 1901, he systematically went through and described and named all of those molecules that make up DNA. When you hear about DNA being the A's, the T's, the C's, and the G's, and it's a really long code of those 4 letters, you can thank Kossel for the names of the A and the T and the C and the G. So he was doing his work on his cow pancreases, and looking at those cow pancreases, he was able to identify and name, initially in 1885, he discovered guanine, that's the G, right, and adenine. So the G and the A these are the rungs on the DNA ladder he identified and named. Now, guanine actually already had a name. He didn't get to come up with that one. Guanine had been previously reported. This molecule had been found and described, specifically in the excrement of seabirds. Have you ever heard of guano? Guanine, yeah. Yeah, this has been a really fascinating topic to research. So guanine came from that, but adenine hadn't been described. He did get to name adenine, and adenine, that name came from the Greek word for pancreas, which I might butcher, but it's something like adenus. So he gave that the name based on the organ of the cow that he was researching. And then a few years later, the slaughterhouse actually started sending him a new type of organ. He started getting cow thymus sent to him. In a butcher shop, they actually call that gullet sweetbread, in case there's people in the room that rings the bell for. But it's a cow thymus. And so he got a bunch of those, and he was doing research on those body parts of the cow, and he discovered, you see it? Thymine, that's the T of DNA. And he also discovered cytosine. Now, this one is a little bit less of a creative term. Really, cytosine was named after a word that means cell, which is cyto. Yeah, kind of boring after some of the other ones. But so he discovered and described those letters. And then also, we can't forget this other molecule called uracil. And uracil is not in DNA. Uracil is actually in that messenger RNA. Messenger RNA does not have thymine in it. This one, everywhere that it would have a thymine, it has a uracil instead. So these are the 5 molecules that Kossel described for the first time. Actually, uracil, I think, was It was discovered by a student in his lab, not him. But this was his big contribution, which is a pretty big contribution to our understanding of DNA, although at the time he had absolutely no idea kind of the impact that these molecules have. So if we fast forward to today, we now know that our DNA, the twisted ladder of DNA, is made up, the rungs of the ladder are made up of these molecules that Kossel discovered, and that they pair together in a very specific way, where G's always pair with C's and A's always pair with T's. And then over on RNA is actually not double-sided like DNA is. It's only one-sided, and there's no T over here. Instead, you have that uracil. So this is what we know now. He could not put all of these pieces together until, Q. Watson and Crick, right? This is a quote from Albrecht Kossel. I think this is in your program as well, but I really like it. It says, "The processes of life are like a drama, and I am studying the actors, not the plot. There are many actors, and it's their characteristics that make up this drama." I really like that, and I like the visual image of DNA being an actor in the drama of life, because it definitely is a key player, for sure. Kossel went on to continue working. He never actually fully retired. He continued to work and make more discoveries. He is buried in Heidelberg, Germany. He traveled to the US one time after he got the Nobel Prize. He traveled here and did a couple of talks at Johns Hopkins and some other places. But that is the story of Albrecht Kossel. So for our next journey down the lane of history, we actually have two individuals that worked together. So this is Edward Tatum and George Beadle. And for this one, we're actually heading to the US. These are two American-born scientists. And what do I need for them? We need bread. And not just any bread. I don't know if you can appreciate what's happening on this bread. It's turned. Not really, it's paint. So that is our prop for this story. So George Beadle was born, let me make sure I get it right. He was born in Nebraska, and Edward Tatum was born in Boulder, Colorado. So they were American-born. They both went off and studied biology and had an interest in genetics, but where they converged, where their stories converged, was at Stanford University. So both of them in their professional career ended up at Stanford together. And together they embarked on a really important set of experiments. And they were not studying cow parts. Their subject of choice was bread mold. I bet their lab smelled great. Right? But they studied bread mold, and you might wonder why. That seems odd. But it actually is kind of an elegant system. It's relatively easy to grow. It's simple. It has a fairly simple life cycle. It reproduces quickly. And the fact that it is simple, you can really grow it on an incredibly simple medium. That's like the food that it would grow on, either in a plate or a test tube. Because bread mold has all of the pathways it needs inside of it to make the building blocks of life, all of the proteins it needs. Pretty much if you put it on a medium that's got some salt and some sugar, it's good to go. It can turn that into anything it needs. And so they had a hypothesis, Beadle and Tatum did, and their hypothesis was a connection. And this gets back to that central dogma thing. They had a hunch that there was a connection between genes, or DNA, and proteins, that somehow those were connected. And that if you found a way to break a gene by cause a change, a mutation, that you could change or break the protein that it is the instruction for, that you could actually see the impact of that. That was their hypothesis. And now looking back, you're like, well, of course, but they had no idea. So they set out to do a series of really kind of elegant experiments to test this hypothesis. Of note, they wouldn't be able to do their work if there hadn't been another scientist that did also win a Nobel Prize, Hermann Muller, who his Nobel Prize was around the discovery that that X-rays cause DNA changes. It causes mutations. Again, we know that well now. That's who discovered that. And so they took advantage of that discovery and said, well, let's see what happens if we intentionally subject our bread mold to X-rays. Let's induce a whole bunch of changes and see what happened. So the experiment was something like this. There is a whole set of red bread mold spores. They irradiated it, and with that, there was a whole set of random genetic changes that happened in some of those spores. They don't control where, it's random, but they are assuming that's happening kind of throughout. And then they take those irradiated bread mold spores, and they plate them out. They put them in tubes, and they grow it up. And they initially put it on, that they're growing it up in tubes that have food, media, that has all the stuff in it, right? That any mold with any kind of mutation can grow on this because they're feeding it everything that it might need. So they get it growing up pretty good. And then they transfer it to a different set of tubes that are what we call simple media. It has nothing but that salt and sugar. So most mold is going to grow on that simple media, because remember, the mold itself has all the mechanisms to make what it needs, unless something broke, right? That for the spores that have a mutation that happened in a critical place, they're not going to grow here when they could grow over here. So those are the ones that they were really interested in following up on and trying to figure out what specifically broke. Can we map an X-ray, a genetic change that happened, to an actually broken mechanism inside of those cells? So they then took these particular spores, and then they did a whole bunch of other experiments, right, where they broke it down even further, and they said, okay, let's grow those same cells on media that only has one extra thing, right? It's simple plus this, or it's simple media plus that. They do that a whole bunch of times over to ultimately ask the question, where's the tube that's rescued by having something added? Because that'll tell you what it is that those cells can't make. Because if you give it that one thing, it's suddenly saved and can grow. So they were able to pinpoint that X-rays would cause genetic changes that would break a very specific process inside of those bread mold cells. And they were able to connect, and they published in 1941, here's a picture from it, this really hallmark paper where they describe what they called the one gene, one enzyme hypothesis, where they said it was an oversimplified hypothesis. Now we know that there's lots of caveats to it, but it largely holds true for a lot of our genetics, where you have a gene that codes for a protein in a 1-to-1 ratio. And they did all of this work with so fewer tools and knowledge that we have today, but really pushed us forward towards that genes code for proteins, and that it's DNA, it's the genes that are the central tenet of that. Once they did this and they published it and they got recognition for it, they actually helped uncover and circle back to this gentleman right here, Sir Archibald Garrod. He was a British physician. He was working way back right around 1900, and he had the same hypothesis. Now, he didn't test it. He couldn't prove it like Beadle and Tatum could, but he was a physician that was caring for patients that had this condition called alkaptonuria. It's a non-fatal condition, but the hallmarks of this condition is that urine, as well as spots in the eyes and even around your ears, that there's these black buildups where urine is black and you get these black spots in your eyes and ears. And patients that have alkaptonuria, the issue is they can't break down a particular product called alkapton, that normally when you don't have this condition, that can be broken down into colorless products. But patients with alkaptonuria, that builds up. And so Dr. Garrod was caring for these patients, and he was watching this flow through families and was saying, this seems to be inherited, right? I think he even hypothesized that I think there's a form of that stuff Mendel was talking about at play here. And that I think there's a change in one of those heredity units that's making this enzyme not work. So he hypothesized it. No one really paid him any attention until Beadle and Tatum did their work, and they said, oh, you know who was right way back then? Dr. Garrod. So Beadle and Tatum, they received the Nobel Prize in 1958 for the discovery that genes act by regulating specific chemical events. They shared this Nobel Prize with another scientist, Joshua Lederberg, who had an entirely different discovery that we don't have time to talk about, but they shared half of this prize together. This is a picture of the telegraph that Edward Tatum got that said, you know, congratulations, you have won the Nobel Prize. So that is Beadle and Tatum. And so we've now gone from, okay, we've identified what DNA is made of. We've identified that, that it's the DNA that is the secret sauce to life, right? Like that's at the central of it. It's not proteins. But proteins are important too, but it's the DNA that controls what those proteins look like and what those proteins do. So that brings us to our next story, which is this gentleman right here. His name's Frederick Sanger. There's something really cool to note about him. What is it? Yeah, incredibly industrious. He has 2 Nobel Prizes in his credit. I'm going to put back my bread mold. Oh, it's actually cardboard. And we're going to pick up our sewer cover. I hope it doesn't fall over. So let's talk about Mr. Frederick Sanger. Fred Sanger, he was from England. He grew up in Gloucestershire. And his father was also, this is a constant theme of a couple of these, his father was a physician. He was very much expected to follow in his dad's footsteps, but he, kind of like Kossel, decided that science and just making discoveries seemed more appealing than being a physician, and so he also went into scientific discovery. Now he went, so looking at the years that he was alive, he was going to college in Europe in the 1930s, which was a tough time in Europe. And so he went to college, and then he also, of note for him, he was raised, his father was a physician, his father was also a devout Quaker, and so he grew up Quaker, and while he did not necessarily follow that through his whole lifetime, during World War II, he was registered as a conscious objector and had a role as an orderly in a local hospital as part of his role in the war. But around that time, he decided to apply to the University of Cambridge to see if he could go get admitted into their PhD program, and he was. And he ended up spending the rest of his educational career, as well as his entire working career, there at the University of Cambridge. When he was in his doctoral program, his project was actually war-related. It was exploring nitrogen in potatoes, is what he did his PhD work on. But what also was happening there during that landscape of time in Europe, there was a group of famous scientists that were based in London, but decided that they wanted to get out of London to a safer location, and they moved to the University of Cambridge. And included in that were some really, some important big-name scientists in England that were interested in proteins. But they were interested in proteins and trying to figure out what proteins are made of and how they behave. And so it was that scientist that kind of converted Sanger over to being interested specifically in proteins and gave him a challenge and said, hey, you should go and you should try to figure out what is in insulin. And so he took up that charge. And what Sanger did was he was really a developer of new methods and technologies. So at the time, there was no good way to just go figure out what a protein was made of. You couldn't do that. And that's what Sanger did. He developed a way to figure out that picture of the protein where it was all of those little balls strung one by one, right? A protein is made up of a whole bunch of amino acids. And so at that point, there was no way to figure out what was that sequence of amino acids. He figured that out. And so he was working in insulin initially, and so he figured out not only a way to sequence proteins, but he figured out the protein sequence of insulin. And that is ultimately what got him his first Nobel Prize, was his work around protein sequencing and insulin. So here's the picture. So he was awarded his first Nobel Prize in 1958. He is the gentleman in the middle. Also in 1958, look who's over there, Beadle and Tatum. This was the same year of Nobel Prizes. We have several of our characters up here. But he got his first Nobel Prize for his work in insulin research and figuring out what is in the protein insulin. So from there, he turned his attention. So he started with figuring out how to sequence proteins. And so he actually made a stepwise approach back where in the next decade of his life, he was really interested in trying to sequence RNA and how to develop a tool and a way to sequence that. He did, but he didn't do it fastest. Somebody else figured it out first and got the Nobel Prize for that. And then he turned his attention to DNA and trying to figure out how can we actually read that genetic code? How can we put those A's, T's, C's, and G's in order? And he did. I'm not going to go into all of the chemistry involved in it. This is a picture from his paper that was published where he said, here's how you can read DNA. And in short, the way that he did it, and you can see all of these little squiggles and lines, is he took a stretch of DNA and found a way that you could essentially kind of read it by making lots of copies, and then you're adding radioactive versions of A's, T's, C's, and G's so that you can see it on one of these gels in the lab, and you have it where it stops at every single letter. And then you put it on the gel, it flows through. The shorter the fragment, the faster it goes through the gel. And so literally you can start at the bottom and just read the DNA up the gel. This is something we still, to this day, call Sanger sequencing. He figured out how to sequence DNA. That's a massive deal. So he figured out how to sequence it, and so then he set out starting to sequence things. So he sequenced this guy right here. This is bacteriophage 5X174. This is the first organism ever sequenced. This particular, so a bacteriophage is a virus that infects bacteria. This particular one was isolated a few decades earlier. The first time this bacteriophage was ever found was in the Paris sewer lines. Promise, we got there, right? But that was the very first organism that was ever published. Now, its DNA ladder is only 5,000 rungs long, Well, humans is 3 billion, so starting kind of small. But his sequencing technique could only sequence 80 at a time. And so that's a lot of experiments, even to do something that is 5,000 letters long. But so he sequenced this bacteriophage, and he got himself another Nobel Prize. So he won the Nobel Prize for Chemistry in 1980. Not just him, there were 2 others that shared in this 1980 Chemistry Prize. Actually, Gilbert here, he got the Nobel Prize as well because he, at the same time, developed another way of sequencing that was incredibly similar, but they were not working together and had no idea. They kind of were doing it in tandem. So he got another Nobel Prize. Sanger is one of 5 people that have ever gotten more than 1 Nobel Prize. He's only one of 3 that have gotten 2 Nobel Prizes in the same category. An incredible legacy he had. Also, as a kind of fun fact, he trained other scientists. He had at least 10 doctoral students that worked in his lab, and 2 of them went on to win Nobel Prizes. Yeah, quite the legacy. Right. So Sanger sequencing is a method of sequencing that quickly got adopted across the globe as the way to do sequencing, and we still use Sanger sequencing today. Now, there's also lots of newer technologies that go a lot faster and a lot cheaper, but there are still reasons that we will do a Sanger sequencing test even today. But Sanger sequencing is the type of sequencing that was behind the Human Genome Project. The big, large-scale, worldwide collaboration of scientists where we sequenced a human for the first time. That's that whole 3 billion letter genome. That was done on Sanger sequencing. That's why it took 10 years, right? And why also we're so glad he did what he did, but we're also really glad we have newer technologies that have come along since then. But yes, a hugely impactful discovery that he made. I also have a really lovely quote that I found that he wrote. Let me read this to you. It says, "Science is not like the Olympic Games or something, where there's a lot of people all trying to win gold medals, and if you don't get a medal, you're nothing. There are actually a lot of people working together and contributing to science, and the science is the important thing." Says the guy who won 2 Nobel Prizes. But no, Frederick Sanger was an incredibly humble man, and he is definitely right that behind every person that we're talking through tonight, there's a lot of people they worked with and a lot of other people that have done amazing science, that winning a Nobel Prize is not what makes you a good scientist or not. But Frederick Sanger was an awful good one. So he spent his whole kind of professional career in Cambridge, and he retired. He actually retired fairly young. So he retired at 65 and decided to go work in the garden and enjoy life. And so he did that, and while he was doing that, Cambridge came to him and said, "Hey, we're building a genome institute, and we want to put your name on it." And the story goes that he said, "Okay, but make it good." And they did. So this is actually a picture of the Wellcome Sanger Institute there in Cambridge. This was one of the many really big driving players of the Human Genome Project, and still a place of active genomic research today. All right, so that was Fred Sanger. And last, but absolutely not least in our journey through genetics history tonight, we have Barbara McClintock. So this is our one female geneticist to highlight this evening, and we're heading back to the US for this one. So Barbara McClintock was born in Connecticut, but at a fairly early age, her father was also a physician. They moved to Brooklyn and started a medical practice, so she spent a lot of her growing up years in New York. An interesting story that when she was born, she was actually born Eleanor McClintock, But at an early age, her parents decided to start calling her Barbara. At least the lore, right, or the stories are that her parents decided that the name Eleanor was too feminine and delicate for Barbara. She was an incredibly independent, I think, strong-willed person from the get-go. But Barbara, and later she officially changed her name to Barbara. So Barbara grew up. She fell in love with science in high school and decided she wanted to go to college. She wanted to go to Cornell, and she wanted to study botany. The story goes something like she told her parents that, and her mom said no. And her mom said, supposedly, did not want her to go to college because she feared it would make her less likely to get married. And she almost didn't go to college, and then her dad enrolled her like right at the last minute. So she goes to college and she studies botany. She takes a genetics class pretty early on and falls in love with genetics. And the instructor was so impressed with her that he started putting her in graduate genetics courses the next year as an undergrad. And so she kind of immediately fell into this combination of plants and DNA. Before, that was really something that was combined a whole lot. We combine that here at HudsonAlpha a lot, but that was not happening in 1930. So she went on and through her studying started, she got her PhD in botany as well, but was infusing it with DNA study all along. She did her research, the thing she was most interested in was maize or corn. She spent nearly her entire career working on the genetics of corn. So here's a picture. This is Barbara as an undergraduate at Cornell. And then this is a picture. So when she was in graduate school, she pulled together a team of corn cytogeneticists. So she said, hey, I want to get a team together, and I want people that know a lot about plants and know a lot about corn, and I want people that know a lot about genetics, because I think we could do some cool stuff together. And she did. So this is Barbara here. Guess who this is? George Beadle. He's the man's everywhere, right? So they actually worked together on some of this same work. But yeah, the guy with the dog, that's George Beadle. But she pulled these teams together, and she really dug into trying to figure out what the genetics of corn looks like. And not just reading, at this point, we're not reading the DNA yet. That's not possible. But trying to look at DNA kind of at that macro, larger scale. And inside our cells, inside our nucleus of our cells, our DNA is really, really, really tightly coiled up into these structures called chromosomes. And so she was the first person to ever map out corn chromosomes. So humans, you and I, inside our cells, we actually have 46 of these guys, 23 pairs. She figured out that corn have 10, and they have 2 pairs of each, kind of like we do. But she was the first person to kind of map this out and create and describe what the chromosomes of corn look like. And then started figuring out where certain traits could be mapped to. So different traits of the corn or the maize, where was it on which chromosome that had the DNA that would be associated with or controlled that particular trait. So she did, she worked in maize her entire career. After she finished schooling in Cornell, she actually got a fellowship from the Guggenheim Foundation to go study in Germany in 1933. Yeah, so she went over and quickly came back. And the scientists she went over there to study with had immigrated to the U.S. too. But she came back to the U.S. And again, the story goes she tried to go back to Cornell, and that Cornell would not hire her as a professor on account of gender. And so she ended up, a colleague got her a connection. What? On account of gender. Sorry. So she ended up, a colleague offered her a position at the University of Missouri. So she went there and started doing a lot of research and had some similar tactics to Beadle and Tatum and their bread mold, maybe because she worked with Beadle, that same concept of using X-rays to induce genetic change and then see what happens, right? And trying to help map where traits are. She started doing a lot of that too with corn. But what she ultimately got the Nobel Prize for was a discovery that she did with corn, but it was a discovery around what we now call jumping genes, and figuring out that the genetics, those chromosomes of the corn, that there were places that would often break and then rejoin somewhere else, that the DNA would move around. And that was a phenomenon that was not known at all at the time. So corn is an interesting subject to study in genetics, and part of why it's, pun intended, ripe for studying genetics is that every single kernel of that corn is its own fertilization event. So on one cob, you've got a whole bunch of siblings. So that's efficient right there. So every kernel is its own fertilization event, and all of those kernels are essentially siblings of one another. And without going too deeply into corn genetics here, that a kernel of corn, you have an embryo down here at the bottom, but then the rest of the corn, the part of it that gives it its color, is this outer layer. And the DNA that's in that outer layer is what's going to dictate the color. So Barbara McClintock figured out that there's a couple of different genes, they're actually on chromosome 9 of the corn, that contribute to what color that kernel is going to be. Now my corn up here is very homogeneous, but hopefully you've seen corn that's not, right? That kernels can come in different colors. And that there are genes, right, that are controlling that. And so she described that there were these two different spots on chromosome 9 that contribute to color. There's one that she, the way she described it was that there's a version that she called C prime, right here it's written as a C with a dash, that if a kernel has that genetic code, it has no color. But if it has a different version of that gene, and she labeled that by just C, there's no prime there, that it would allow for color to happen, but it doesn't dictate what color it is. It just says you can have color. And then there was a separate area of the genetic information that was just a little bit further down that chromosome that would dictate what color that kernel would be if color was allowed. So there was a version of this gene that she labeled capital BZ, that if that was present, that the kernel would be purple. But if it was a different version, purple, if it was a different version and it was lowercase bz, then it would be brown. And so you could have a cob, right, where you've got some that are colorless, some purple, some brown, right, all on that same cob. But you've got this interaction of these two different genes that are contributing to that color. And the ones that say dominant, so the C prime and the capital BZ, those are the dominant versions of that gene, which means if that is present at all, it's going to be in charge, right? It's going to say what happens. For a kernel to have color, it can't have any of this dominant version. All of its versions of the gene would need to be that recessive version. Okay? And then she discovered that there was another place also on that same chromosome that isn't controlling color. This was a location in the DNA that she labeled DS, which that stood for dissociation. And it was a place where she was recognizing that DNA often broke, that it was an area of common breakage that was near these other two genes. And so the way this played out in the field was if you had kernels and you had, the egg and sperm, right, the male and female gametes coming together, where you had one that was the recessive versions with no dissociation, and then you had another version coming in that was the dominant versions and dissociation, you would expect that the offspring would be dominant because one of its parents had these dominant versions of the gene, and so that would take over. But what she actually saw was corn that kind of looked like this, right? That had these speckles, that had spots, that were not purely colorless. And so her hypothesis that then she went through and proved was that there was these chromosome breaks that were occurring and were allowing those kernels to lose those dominant versions of the genes, where all that was left was the recessive. And you would end up with these speckles because sometimes it would happen a little bit further along development, so only parts of the resulting kernel would have that change in it. This was a big deal, that genes and DNA can move around, right? That it's not totally static. And so she named these transposable elements, or jumping genes, and that there were DNA sequences that can move from one location to another. And then, so she made that discovery. People didn't believe her a ton initially. It took a few decades and a few other people to replicate her findings in some other organisms for them to go, oh, yeah, that's a thing. And not just a little thing. This happens in lots of organisms. A huge portion of the human genome is the result of DNA that has moved around over the history of time. And so this is a much larger phenomenon than she even knew at the time. So she did that work in the 1950s, but she was finally awarded the Nobel Prize in 1983 for her discovery of genetic transposition. And a fun fact about Barbara McClintock, she still remains the only woman with an unshared Nobel Prize in physiology and medicine. She was wicked smart. I want to read you, as we're kind of closing, somewhere in my notes here. There's this great quote somewhere. So George Beadle, he's a constant thread through many of these stories. So George Beadle actually hired her and brought her over to help them with some of the bread mold work. The scientific name is Neurospora crassa. But he said, hey, she's super smart. Let's bring her out here to Stanford and have her help us with some of the genetic mapping stuff in the bread mold. So she did. But here's what Beadle had to say about Barbara. He said, Barbara, in 2 months at Stanford, did more to clean up the cytology of Neurospora than all other cytological geneticists had done in all previous time on all forms of mold. It's high praise. But she was an incredibly industrious, brilliant scientist who, in fact, did never marry. It's fine. So those are 4 of so many stories we could tell of scientists that have gone on past, as well as current scientists that are out there today, currently winning Nobel Prizes, or as Fred Sanger alluded to, amazing scientists doing work that maybe aren't winning Nobel Prizes, but that's not the mark of what makes a good scientist. But there are so many countless others. I want to specifically point to this gentleman who I didn't tell his story because I would rather y'all come and hear his story from him. So Harold Varmus is a Nobel Prize winning scientist. He won the Nobel Prize in 1989 in physiology or medicine. He also previously directed the NIH, as well as the National Cancer Institute. His discovery that won the Nobel Prize was in oncogenes, so genes that help drive cancer. But he's going to be at HudsonAlpha in this room or that room on November 19th. So that's a point of reference and a welcome and an invitation to come back and actually hear a Nobel Prize winner, not me talking about Nobel Prize winners. But kind of in closing, as I think about the Nobel Prize winners that we've talked about tonight, as well as the amazingly brilliant scientists that are working across the globe, including on our campus, what they have in common. What is it that makes a scientist a great scientist and someone that can really catapult the field forward? And for me, the thing that keeps coming back to is curiosity and that desire to ask questions, but not just ask questions, but figure out how to go get the answer for those questions. And not necessarily just take what everybody else thinks the answer might be, but actually try to go get the answer for yourself and be willing to, not accept what everybody thinks is going on with the world around us, but find those answers for themselves. And as we reflect on the people that we've heard about tonight and the work that happens here on HudsonAlpha's campus every day, we couldn't do what we do without all of these scientists that have gone on before. Every day, there are genomes of organisms being sequenced at HudsonAlpha that we rely on. Kossel's discovery about what DNA is made of, Watson and Crick's discovery about what DNA looks like, Sanger's discovery of how do we actually read DNA, as well as the improvements since then. And many of these scientists' ability to try to map, and even our scientists here at HudsonAlpha continuing to try to map traits, disease risk, those types of things to specific locations in the genetic code, is work that we continue to do that is very much heavily based on the discoveries that were made by previous greats. So in conclusion, I can't give anyone an actual Nobel Prize tonight, but one of you that is sitting at a table does have a HudsonAlpha U prize attached to the table underneath in front of you. If you want to kind of reach to see if there's paper taped under the table in front of you. Anybody? Oh, we found it. So, this is not almost $1 million in cash prize. Again, this is not a Nobel Prize. But what I am, Susan, will you come grab this? But what I am going to give you is a HudsonAlpha U cup. Yes, congratulations. And you too could have a HudsonAlpha U cup. This is also a little bit of a shameless plug that our education team has an online store where you can not only get HudsonAlpha U stuff, but other stuff as well. And the way that is built is that proceeds from those sales go back to help support the work we do with students and teachers and everything else we do in education. So that is a shameless plug, but also a chance to give a cup away. Yes, in closing, here on the back of your program, there's a whole list of things, not just HudsonAlpha U, but other events that you can come to here. In terms of HudsonAlpha U, we've got an event in September all about family health history. What can your family history, we did ancestry last fall, but this is really what can your family history tell you about your health and disease risk, and what can't it tell you? So we'll dig into that in September together. And then in November, we've got, we started doing this last year. It's an alumni appreciation event. It's a reception for people who are members of the Alumni Association. That's an affinity group where you also are, it's to say thank you to our supporters in Hudson Alpha Education, where you can come and get kind of a behind the scenes look at one of our programs or one of our activities, get to play student, and also just let us say a big thank you. So that's what's coming up later this year. With that, thank you for being here tonight and going with me on this journey through the past. I hope it was as fun for you as it was for me to do the research and get to share the stories. But y'all have a great evening, and we'll see you soon.